Okay. I think we'll make a start for it while the computer's loading. So, genetics, but I'm just waiting for these guys to shut up so I can talk.
Guys.
But I'm just waiting for it. Are you gonna listen or not?
Yes. Guys.
Okay, so in the last lecture we talked about DNA replication. Remember, before cell division the DNA needs to be replicated, and there's certain enzymes involved in that. So if you remember that DNA is a double helix, so it has two strands complementary to each other. So A to T, G to C, and each one of those strands is used as a template to make a new strand during DNA replication. So in this lecture we're going to talk about transcription. How do we convert the DNA code into RNA? So if we have a look at, I'll give you an example how the genetic code works, okay? So if we have DNA, let's use another pen to make it clear. So let's say I, T, C, G, I, C, okay? And you want to convert that to RNA. So you have two strands, okay? Remember, DNA is double strand, so the opposite strand would be T, I, G, C, T, G, okay? So DNA is double stranded. Now, you know how we separated the strands for DNA replication? We also have to separate the strands for transcription, okay? So in transcription, there's an enzyme which is responsible for making the RNA. So remember in replication we had DNA polymerase?
Yes.
DNA polymerase makes a copy of the DNA. So to make a copy of the, make an RNA from DNA, we have another enzyme called RNA polymerase. So in transcription we use RNA. So this enzyme is responsible for converting the DNA to RNA, okay? Now, during transcription, what's happened is that, let me just go to this one. If you look at the gene, you know, remember the gene, the basic unit of, the functional unit of DNA?
Yeah.
From the last lecture? And we talked about gene expression, which is expressing the gene as a RNA or finally as a protein. And we had 20,000 different genes.
Mm-hmm.
Yes?
So we use the DNA polymerase to bind the parent strand with the new strand.
So the DNA polymerase makes a new strand from the original template strand. So this, when you separate, that's in DNA replication, okay?
Yes.
So this is transcription.
So what?
We use RNA polymerase, different enzyme.
To bind the new one.
So I'm going to talk about that now, okay? Before I do that, I need to explain the structure of the gene, okay? We talked about, you know, a gene is a functional unit of DNA. Now, a gene has a particular structure. So the RNA polymerase can't just, it has to look for certain sequences to identify the gene, okay? Unique sequences on every gene point to where the transcription needs to start, where it needs to stop, okay? So a gene has a particular structure, okay? If we assume this is a gene, okay? it's not, the genes are not that short, okay? So they're much longer than that. A gene usually have what we call a sequence. I'm not gonna write this DNA sequence there, okay? I'm just gonna use drawing here, little pictures here to annotate the different structures of the gene, okay? So it will have a sequence here, which we call the promoter. Now this is a DNA sequence where it actually recruits the RNA polymerase, okay? So the RNA polymerase will bind to this region in the promoter region. The promoter region, without the promoter, you will not get gene expression. So the promoter is important for gene expression. Let me just log on to here. So all genes have a promoter. So the promoter is the start of transcription, okay? Then you have the sequence that codes for that particular gene. So the coding sequence, okay? All right, so this could be the coding sequence for insulin, okay, for example. Remember, did I go through with you with the genetic code, the codon table yet, or not?
Went through it quickly.
Okay, so I just wait for this to load, then we can. So then you have the sequence which actually codes for the protein, okay? So in this case, the insulin, hormone insulin, which is a protein. In the end, you have another sequence, okay? We call this the terminator. So the terminator, it acts as a end of transcription, okay? Okay, so every gene has a promoter, then it's got the coding sequence, okay? Then it has a terminator. So this promoter is where the RNA polymerase binds and starts transcription. You'll transcribe that DNA, okay, into messenger RNA, okay, until it reaches this sequence here on the gene, which is the terminator, and then it will stop transcription, okay? So, but this is basically what's happening with converting the DNA to RNA. Yes?
So what's the function of the terminator?
Terminator to stop transcription, okay? Once the gene has been transcribed, okay, we don't want this polymerase going, transcribing the whole genome.
Yeah.
So there has to be an end to it, okay? So the terminator ends transcription. Now, this process can happen for a particular gene. It will happen many times. So you'll get RNA polymerase binding, transcribing, and it will come again and keep doing it, depending on the amount of RNA that is required, okay? So, for example, if a lot of insulin is required, you get lots of transcript produced, okay? If insulin is not required, it'll inhibit the transcription, okay? Because what we're not talking about here is that in the promoter region, there is certain other regions which can help to increase transcription or decrease it. It can regulate gene expression in the promoter region. Okay, let me just so what happens is that RNA polymerase, this protein RNA polymerase, will come and bind to the promoter region, okay? once it binds to it, it unwinds the double strand, okay? Just like the helicase unwound the DNA, it will unwind the DNA, and it will start transcription. So in the promoter region, so not all of this gene is transcribed, okay? So there is, in the promoter region, there will be a start of transcription, okay? So start of transcription will be somewhere here. So not all of the promoter is transcribed by the RNA polymerase. These sequences are important for the binding of RNA polymerase, okay? So it won't transcribe the whole promoter, it will have a region within the promoter or just after the promoter that is the start of transcription, okay? So what will happen is that the RNA polymerase, when it binds to this promoter, it'll start transcribing the DNA into RNA from the point of where the start of transcription is, okay? So you'll get the RNA molecule forming, it'll transcribe it, okay? So you got the gene, all right? And then in the terminator region, there is a region where there is this end of transcription, okay? So it won't transcribe the whole terminator, okay? So this is now what we call the
Transcription.
the primary RNA transcript, okay? So in the book, in your lecture, it says RNA primary transcript, okay? So this is the transcript that is coming off. So now we converted this sequence. Now what's happening is when these two strands are opened, okay? So the RNA polymerase will start to convert, so it'll use only one strand, okay? So we call the sense strand, and this is the anti-sense, okay? So the two strands are complementary to each other, okay? So when the RNA polymerase opens it up, it'll start adding, it'll use this anti-sense, okay, as a template, because we need to get this code, okay? So we have to use this strand, correct? To get that code, we need to use this, because it will give us the complementary. So what will get here? We got a T, what will be? I. We got I, what will be?
T.
U. We're making RNA, okay? Uracil. So C, G, I, C. So this is the RNA molecule. Now I'm only showing a little bit, but this will be a large molecule, okay? So we've made the RNA, okay? So this is the RNA, or the primary RNA transcript that is coming off that gene, okay? I don't have my USB with me, so I have to log on to that. Does anybody have a USB? Can I borrow it for a second? Instead of going through here, I can just copy from my laptop. Thank you. So when this primary RNA transcript is made, now eukaryotic organisms have a different gene structure to prokaryotic organisms, okay? So what we find in the eukaryotic organisms is that the gene contains two types of sequence, okay? What we call introns and exons, okay? Have you heard of these terms?
No.
So there's two types of sequences. The introns, they're like, when they were first discovered, they were thought to be like junk DNA, okay? Because they didn't code for the actual protein. But later on we found out that they're not actually junk DNA, they have functional sequences within them. They're important for regulating the expression of the gene, sometimes also containing, some introns contain other functional RNAs, okay? So they were first thought to be like that, but they have function, okay? All right, and the exon is the coding sequence, okay? So this is what will give us the sequence that is translated, the translated sequence. Now, what we find is when we look at the structure of a gene, we find the introns are located within. Sometimes, not always, okay? So you find that you have, when you look at a gene, you have exon, intron, then another exon, then another intron. You know, I'm just showing two different genes have different number of introns and exons, okay? So this is just an example. Now, the coding sequence lies here in the exons. So the ribosomes, if they read this transcript, okay, they'll make mistakes in translation because there's an interrupting DNA of introns, okay? Or I should say RNA, okay? Now when it starts, the ribosomes, they read the sequence in triplicates, okay? Which we call codons, okay? Have you heard of the word codons?
Yeah.
So this, after transcription, we get translation. So every 3 code is read, and every 3 of the sequences code for 1 amino acid. Now, hopefully we got the computer running so we can. You wipe this off? Okay?
Yeah.
We'll go to the lecture notes. So, if you have a look at the structure here, this is a particular RNA molecule, okay? Say it's that molecule that we were talking about. Now, you see that there is, it has these introns and exons. Now, this is the primary transcript, okay? This is what was transcribed initially from the DNA into RNA. Because it contains these extra sequences, these introns, which are not coding for amino acids, okay? So introns don't code for amino acids. Exons code for amino acids, okay? They need to be removed before translation, okay? So the introns are removed before translation. So there's enzymes involved that recognize the introns, okay? And cut them out, okay? So the introns are cut out and the exons are joined together, okay? To give this what we call the final mRNA transcript, okay? Or mature.
Who is responsible for removing the introns?
Enzymes, there's actually a wide range of enzymes. It's actually called the spliceosome, okay? All right?
All right.
There's several enzymes that are involved that actually they can recognize particular sequences in the junction of introns and exons, okay? And they know where the introns are. They will slice them out, okay?
Yeah.
And join the exons together to form the final or mature mRNA transcript, okay?
And where does the introns go?
Well, they disintegrate, okay? If they have some functional, they will be used further on, okay, for other. Sometimes they can code for what we call microRNAs, which we're not going to deal with, okay? Okay, so the introns need to be removed. They don't code for protein. The exons are joined together, and this is what is translated, okay, by the ribosomes. So I went through this, transcription is, this is the message, okay, from a single gene that is converted from DNA to messenger RNA, okay, and this is what is carried to the cytoplasm, okay. So transcription happens in the nucleus, all right, because that's where the DNA is. And it's called messenger RNA for a reason, because it takes the message from the nucleus to the cytoplasm. I already talked about this before. I'll just go through it briefly again. So remember, the promoter is where the transcription begins, and the terminator is where it stops, okay? The RNA polymerase is the one responsible, so it binds to the promoter, okay? And then unwinds the DNA. And then RNA nucleotides are attached to the complementary DNA. So remember that when we have A, you get U. If you have T, you get A. Then you have C, you get G, and so on, okay? So the RNA polymerase, it will start adding the RNA nucleotides and create a RNA molecule. Yes?
So the ATC sequence are from DNA or from RNA?
This is the DNA, okay? Because in DNA, we find T,
Thymine.
okay? In RNA, it's uracil, okay? And the sugars are different. One is the ribose sugar and one is the deoxyribose, okay? So deoxyribose, ribose. And then this process will continue. The RNA polymerase will continue to add RNA nucleotides complementary to the DNA, okay, until it reaches the terminator, and then you make the primary RNA transcript, okay. So then remember that the primary RNA transcript contains exons and introns, so there has to be some editing of the primary transcript to get the mature or final RNA transcript. So as I mentioned before, the spliceosome, this is a set of many enzymes, the spliceosome machinery. It has enzymes to identify the introns based on sequences, and then enzymes to cut it out and join them back together to get the final mature or final mRNA molecule, okay? Now, sometimes a gene can give two different variations of the transcript, okay? And the introns can help in that as well, okay? So, what we call alternative splicing, okay? So the introns can also allow for the exons to be joined in different ways. What we term, we get now we get variations of the transcript, because what happens is sometimes, you know, it might splice out, you know, 3 of the introns and leave a portion of the final intron, okay? So that means we have now extra sequence in the code, okay? So this can give us slightly different versions of the protein, okay? So the introns can help to give slightly different version of the proteins by what we call alternative splicing, or alternative splicing that can give us mRNA transcript variants, okay? So we can get variants of the mRNA transcript by, for example, not splicing out one of the introns, okay? Or splicing it in a different place within the intron, okay? So what we do is we get different versions of the same transcript. We call that mRNA transcript variants, okay? And some genes do that, okay? Do you understand how that's happening? So depending on where the intron is spliced, we can get different transcript versions. So this is what it's saying here. You know, the introns can allow exon fragments to join in different ways, so we can get different versions. So they're slightly different mRNA molecules, so we get different versions of the protein. Okay. All right, so if we have a look at the genetic code, as I mentioned, we got the triplicate codes, okay? So, for example, these 3 nucleotides, mRNA nucleotides, will code for 1 amino acid, okay? So, in the next one, and so on, okay? So, each one of these triplicate nucleotides, or codons we like to call them, all right? So they're known as codons. They code for 1 amino acid. Now, if we have a look at the codon table here, this will be called the codon table, or the genetic code, okay? It's universal to all organisms, so bacteria, plants, animals, fungi, protists, they all have the same, they use the same genetic code, okay? If you have a look at here, how many nucleotides do we have in DNA? How many nucleotides make up DNA? 4, huh? So, I, T, G, C. So if we put that, we convert that into mRNA, just so we can see it in this. So we have U, I, C, G, okay? So this is the mRNA. This is the codon table, okay, or the genetic code. So what it shows the first position on this way, okay? So position 1. So U, C, I, G. So you see, and then it shows the second position, so second position, all right, and then third position here, okay? So if we have a look at this, we have a look at these 4 nucleotides. When we do the different combinations, we get 64 different combinations. So there's 64 different combinations when we combine these 4 nucleotides in different ways. Okay? Do you understand that? You know the different combinations? Anybody doesn't understand that? Okay. You got 4 base nucleotides, okay? Let's take and make one combination. Let's take, you say the first combination is this, this and that. So we got U, C, G. Oh, oops. Did I put it? U, C, G. Alright, let's make another combination using C, A, and G. C, A, G, okay? And we can use this to make different combination, but it has to be 3 letter, okay? If we do all the combination using these 4 in different ways, we get 64.
Yes.
You got that?
Yeah.
Yeah.
Is it different if we change the places? If we
Yeah.
say C, U,
Yeah, we
G?
can make any combination using these 4.
No, I mean, now we have UCG.
Yeah.
Is it different for GCU?
So if you do, if you have G, C, U, it'll be a different because it's one direction. Okay? You can't read it like that. RNA is read in one direction by the ribosome. So this is different. Now if you have a look at here, we only have 20 amino acids and you have 64 different combinations. Well, the cell knows about this, and so it uses several different combinations for one amino acid, okay? So if you have a look at here, for leucine, okay, we have 4 different combinations. Unless there is anywhere, somewhere, okay. So 4 different combinations, okay? Now, as we had for transcription a start and stop site in the sequence, we also have start and stop for translation when this code is translated into protein. So we always have the AUG, which is a methionine. This is the start of translation, okay? This is where the ribosomes will start translation at a methionine, AUG. And it will stop translation, so it'll read the transcript and it'll reach the end of the coding sequence, okay? And this is where the stop codon is, okay? So these don't code for any amino acids. They just tell the ribosome that this is the end of translation, okay? Termination of translation. So we have 1 start and 3 stop codons. And several different combinations can code for 1 amino acid, okay? Because we only have 20, and the IUG is the only one that codes for the start, or it can also code for methionine within the protein, all right? Let me just go back to here. So if we have a look at the final transcript that we got after so the ribosomes will use this as a template, the final, okay? So this is the template for the ribosomes. Alright? So the final mRNA transcript that we got here will be a template for ribosomes, and it'll read that. So what it happens is ribosomes will begin translation from this start codon, okay? So this is where the triplicate codons are converted into or translated into amino acids by the ribosomes, which we'll talk about tomorrow. Okay, so translation. And so the codon table, as I said, is 20 amino acids. Okay, 64 different combinations. So that's why several different codons can code for the same amino acid. Okay. Any questions on that? I think I explained that on the table there before. Yeah. All right, so let's have a look at this activity. So when we look at this table, how many start? One start.
3 stop.
Okay. How many codons are there for serine?
4.
4?
Yeah.
Who thinks there's 4?
6.
6?
Yeah, 6.
There's 6, okay?
Yeah.
Because we find here there is 2 more, sorry. So 4 plus 2, 6.
The
Okay.
only one is 6.
Huh?
It's the only one is 6.
When what?
No, we have the lace.
I think that's the one.
Leucine have 6 also.
Leucine?
Yes.
Yeah, here. Okay.
Also the arginine on the right with the serine. 2 and up of it.
Yeah. So,
So every protein have 6.
no. Some.
Depends.
Okay. So if you have a look at violin, I think it's 4. No more violins anyway. Some have only
2, 17.
Okay. So if you have a look at lysine, it's 2.
Also the cytine.
Where is it?
Up on the right.
Cysteine, you mean?
Cysteine.
So, or it can have 1. The start codon also codes for methionine, okay? In the middle of the protein there might be a methionine amino acid that needs to be added, so it will be this code, okay? So this codes for the start and also for methionine, okay? So serine is 6, huh? Okay. So what's the amino acid sequence of this? Come here.
What?
Come up here. Use that table to convert this to amino acids, okay? And you can use the 3-letter code, okay? So find the sequences on there and tell me, translate that sequence into amino acids. Yeah, so in the start you will have a methionine, so put methionine. Put the full METH because in
Yes, I was looking for.
yeah. Just put M, it's written there. MET, okay? So you know whatever the amino acid is, it's written there. Next to it. Okay, thank you. So IUG, methionine, UAU, tyrosine, UAU. Where is it?
Up there.
There it is, tyrosine, isoleucine, IUU, CIU, okay, histidine, and then you got the stop. Come here.
Doctor, could you explain?
Huh?
Could you explain?
Explain. So what happened is there was a mutation. Say a mutation that changed this A to C. Okay? So now from UAU, the sequence is UCU. Okay? In this codon. Okay? So what does that cause? It changes the amino acid sequence, just one base pair. Okay? So instead of tyrosine, we have serine. Okay?
Yeah.
What if there's a mutation in CAU? Okay? So if there's a mutation that changes this to C.
C.
Yeah. So what sort of, will that mutation change the amino acid sequence? No. So some mutations change amino acid sequence while others don't change. Okay. So this is a silent mutation. You know, it doesn't affect the end product. So there is a mutation. Okay. But it didn't affect the amino acid sequence. Okay.
We will have the same amino acid.
Yes, it will have the same amino acid sequence, even though the DNA has changed, okay?
Yeah.
It had no effect on the amino acid. So you understand how this codon table works?
Yes.